Okay. So the transport of multiple molecules. So eukaryotic cells need to move and take in larger molecules and particles. So the transport of large molecules, particles, or cells across the cell membrane is called bulk transport. So bulk transport requires energy. So we must understand that this cell membrane is a selective barrier, is a discriminatory. It chooses and allows who's welcome to go inside the cell, who can go outside the cell. It's like the school here, you come in with an ID. If you don't have an ID, you cannot come in unless you have a pass, stuff like that. So the cell membrane is also like that. Cell membrane is very dynamic. Your cell membrane at any moment in time will not be looking the same as it would in an hour later. If you take a meal, if you're starved, if you're exercising. So it's changing on demand. So your plasma membrane is like the universe, like space. You take a photo with the Hubble telescope of the space now. Then the same place, you leave the telescope on the same location. You take another snapshot, it will not look the same because the stars are moving, the planets are moving, so you will get something else. Same thing. Your plasma membrane is changing. There's different channels that are made by the minute, depending on what's happening in your body. So now your cells talk to each other. They communicate with one another. There must be an ability to why are we transporting across the cell membrane? Why are we transporting across the membrane? Well, that depends on what are we transporting across the membrane. We transport electrolytes, and you will see that's important for the nervous system because you will see that sodium flowing in will cause depolarization of the cell membrane and changing the voltage of the cell. The extracellular movement, the movement of potassium from inside the cell, the efflux from inside the cell to the outside of the cell, that will cause repolarization of the cell membrane. So the movement of the electrolytes are important for communication. What else do we need to move across the membrane that is not an electrolyte, that is not sodium or potassium or calcium? What do we need to move across the cell membrane? How about water? The cell needs water. So there are water channels called aquaporins. Aquaporins. These aquaporins allow for the entry of water through the cell membrane because water is polar. Water does not just diffuse down the membrane. You have to understand that just because there's an there's a gradient, there's a diffusion gradient. Just because there is a lot of water outside the cell, it does not mean that the water can just flow in. Water needs a tunnel to go through, um, known as the um, aquaporin. And you know that now the cell is moving both ways. So we're bringing things inside the cell and we are sending things outside the cell. So from the name, exocytosis and endocytosis. So from the name, endo means bringing in or sending out. It means bringing in. Exocytosis, does it mean bringing in or sending out? It means sending out. Excellent. Now the problem is cytosis means bubble, vesicle. So endocytosis, I bring in through a bubble, through a vesicle. Exocytosis, I send out through a bubble, through a vesicle. So when a channel open and you have sodium influx, is that endocytosis? Is that endocytosis when sodium flow in the cell? No, because it's not flowing through a bubble. I just told you that if it has to be endocytosis, it's not enough that it's going in. It has to go in through a bubble. It has to go through a vesicle. That's known as endocytosis. So calcium, potassium, and sodium do not come inside with a vesicle. They come inside with a channel, with a tunnel. So that's an example of an influx. But endocytosis is when you come in through a package, through a vesicle. So endocytosis is a type of active transport, is a type of active transport that moves particles into the cell through a vesicle, through a bubble. So phagocytosis. Phago, what does phago mean? Phago means eating, eating. Phago means eating. Cytosis, you know what cyto is. Cyto means cell. So that's why endocytosis, bringing inside the cell with a vesicle. Exocytosis, sending outside the cell with a vesicle. So there's phagocytosis. That is when your macrophages and neutrophils gallop up, swallow, and bring inside, bring intracellularly some dangerous microbacteria, virus, parasite, fungi, worm that has to be brought inwardly so it can be destroyed. So that's one example, phagocytosis. There is a receptor-mediated endocytosis. That's how cholesterol makes it inside your cell. So there are receptors that are on the surface, on the plasma membrane of the cell, and then cholesterol would bind to the receptor. Once it binds to the receptor, then it's going to be endocytosed, brought into a vesicle intracellularly. Then that vesicle, that balloon, that bubble with cholesterol inside it is going to be taken into a storage site where we're going to store this cholesterol that is put inside it. Exocytosis is a type of active transport. Is a type of active transport that expels material, but also in a vesicle, in a vesicular, in a bubble, in a balloon shape. Exocytosis. Okay, now this is phagocytosis. That's what I was talking about. You see in phagocytosis, this is the cell membrane. Now the bubble, is the bubble moving in or is the bubble getting out? It's moving in. The vesicle is moving inwardly. And this is the particle. The particle could be food. The particle could be a microbe. So we bring it inside. If you're going out, please don't shut the door all the way. Just close it, but leave it a little bit open. So in phagocytosis, there's cell eating, and that is the process by which the cell takes in larger particles, including other cells and cell debris. Neutrophils are a type of white blood cell that removes microorganisms from the body using phagocytosis. So the neutrophil uses pseudopods. These are membrane extensions to surround and engulf the microorganisms. It uses pseudopods. These are like appendages that allow for the movement, of the cell. So there we go. Now that is receptor mediated. You see what I was talking about? You see now we weren't brought in like phagocytosis. Does phagocytosis need a receptor? Does phagocytosis need a receptor like here? That's the receptor. You see, when we bind to the receptor here, that's receptor-mediated endocytosis. But when you look here, there is no receptor. So that's phagocytosis does not require a receptor. That's why we said we have different types. Phagocytosis, receptor-mediated endocytosis, then there's pinocytosis. There you go. That is an example of phagocytosis. This is an example of clathrin-coated pit. This clathrin-coated pit would bind to a receptor. So if we don't bind to the receptor, you're not going to be engulfed inwardly. You see, we go through steps of receptor binding, then there is actin-driven protrusion of the cell membrane, then there is coupling actin to the surface, then there is membrane delivery, and then there is the membrane fission. So now you have the vesicle is going to be endocytosed and brought inwardly inside the cell. Okay, receptor-mediated endocytosis. Here we have the cells use a receptor-mediated endocytosis. We talked about that. You have to bind the receptor. Remember what we just mentioned, the clathrin-coated pit? The clathrin-coated pit is going to bind this material, this substance that I want to bring inwardly, that I want to bring inside. Because remember, All of this is endocytosis, but we are coming in the cell. How are we coming in the cell? Are we coming in by phagocytosis, cell swallowing? Are we coming in by receptor mediated and a clathrin coated pit as shown here? That's a protein which helps, clathrin is a protein which helps stabilize the cell membrane and is often used. Okay, this is a clathrin-dependent endocytosis. So in clathrin-dependent endocytosis, you would see that this you see this protein receptor. Where is the receptor found? The receptor is found at the surface of the plasma membrane. So if you want to come in, you have to bind to that receptor. What is the receptor made up of? This receptor that is going to cause this clathrin coated pin, this clathrin coated pit, in order to bring inwardly, this protein. If you want to bring it inwardly, where do we have to bind? Where do we have to bind if we want to be endocytosed internally through a clathrin-dependent endocytosis? Where do you have to bind? You have to bind to the receptor. You see this receptor here? If you don't bind the receptor, you see, this is what you want to bring in. You see this circle? If you do not bind the receptor, you cannot be brought inwardly. That's why they call it clathrin dependent. So the clathrin is not the receptor. The clathrin is the protein that is going to come later to engulf and surround this vesicle and bring it inward. But you first bind the receptor. Then after you bind the receptor, you see clathrin is going to come. You see clathrin is here in blue. So you first bind the receptor, then after you bind the receptor, clathrin is going to bind you, and then you're going to bring everything inwardly. Does that make sense? Okay. Now this is pinocytosis. Pinocytosis is a variation of endocytosis where a cell takes in water. So pinocytosis is very similar to phagocytosis because here pinocytosis is cell drinking while phagocytosis is cell eating. So you remember it like that. We bring in the cell endocytosis through different ways. You bring the cell inward, you bring inwardly endocytosis through either phagocytosis, and that is known as, what is phagocytosis known as? Cell eating. Cell eating. And then you have pino. It's very interesting that they all begin with a P. Cell eating and pinocytosis. That is known as, pinocytosis is cell drinking. Cell drinking. Phagocytosis, cell eating, pinocytosis, cell drinking, and lastly, there is the receptor mediated pinocytosis. No, not pinocytosis. Receptor mediated clathrin coated pit. Clathrin coated pit. Are we clear? Clathrin coated pit, and that is receptor mediated clathrin coated pit. Okay. So pinocytosis is cell drinking, where phagocytosis is you bring in big molecule, big size molecule, while cell drinking is small size molecules. Big size molecule versus small size molecule. So pinocytosis results in smaller vesicles than phagocytosis. So the vesicle does not merge with the lysosome. So the lysosome is going to merge when you have the phagocytosis, the cell eating. Now there is podocytosis. In podocytosis, you have small molecules and ions are taken up by the vesicle and are deposited directly in the cytoplasm. That uses a protein known as caveolin. Caveolin is a coding protein with a similar function, than to the calathin. So calathin helps in receptor-mediated endocytosis. You see, calathin is helping in receptor-mediated endocytosis, and that uses but caveolin is important in the so caveolin is important for coding a protein with the podocytosis, bringing in the really tiny small molecules and depositing them inside the cytoplasm. Now, if you see transcytosis, what is transcytosis? Trans means right across. So we call it, you see the arrow? I like the arrow here. You see where my mouse is pointing, meaning transcytosis. So we are crossing the bridge. We are crossing the water. We're going from one direction to the other. Now we finish with the endos. We finish with coming in. We finish with coming inwardly. So how many different ways can we come inside the cell? Did we already mention? We mentioned. So what different kinds of endocytosis we talked about? I had them on the board. There's 4 of them. What are they? Did we mention? So phagocytosis, that is when I bring in larger molecules and then I fuse with the lysosome so we can destroy them. Because the macrophage, an example of that is the macrophage, the neutrophil, the eosinophil, swallowing the parasite, swallowing the bacteria, swallowing the virus, and the lysosome fusing with that phagolysosome to chop it up and destroy it. But that's one type, phagocytosis. But we're still coming in. Give me another way that I can come in the cell. What is that? I'm sorry? Pinocytosis. What's pinocytosis? Cell drinking, which means bringing in really small molecules. Phagocytosis, you bring huge molecules and you fuse with the lysosome. You fuse with the lysosome. That is very important. Now, what's the other part? The other part is we have pinocytosis, we have phagocytosis, we still have 2 more. What are they? Yeah, excellent. Receptor-mediated endocytosis, and it requires a protein with a Th, with a C and TH. What is that called? Yeah, calathin. This calathin-coated pit bringing in these large molecules. And lastly, the one where I said cross, transcytosis. There's a protein that involves caveolin. So both of them begin with a C also. Calathin begins with a C, and caveolin begins with a C. But calathin is for calathin-coated pit, bringing in into the inside of the cell, like for cholesterol. And then you have, besides calathin, what do you have? Caveolin, that brings in this, what is the movement known as? So Pato. You see here, if you look up here, please, you see podocytosis, and you have a caveolin bringing in this molecule. So you have to go home and read that. You have to go home and look at these slides. Okay, so now we're done with endo. We're done with bringing in material. Now we have to talk about material that we're going to send out. Okay? So we have exocytosis. Exocytosis is sending out. What's the similarity between endocytosis and exocytosis? Can you please think about that? What's the similarity between endocytosis and exocytosis? Now, my question, I did not ask you what's the difference between endocytosis and exocytosis. Because obviously from the name endo, one bringing stuff in, exo, one sending stuff out. I never asked the difference. I asked the similarity. What is the similarity between them? Yes, Tarata. You went like that. No, yes, anybody. I never said that word one time since I began this lecture. Actually,
Where's the transportation of like?
Firing energy? No.
did we mention energy there? When we were talking about phagocytosis coming in, podocytosis, small molecules, pinocytosis, receptor class. It's the bubble, the vesicle. Both of them, you're not coming in like water, you're rushing in, or with like some ion going through a channel. No, you're bag. You are in a bag, in a bubble, in a vesicle, in a balloon. But one is the balloon coming in, the other one, the balloon going out. So do you see? Do you see? Please look at exocytosis. You see, this is the problem because you're not able to look where my mouse is pointing. So in exocytosis, you see, the bubble is going to come fuse. But in endocytosis, it came in. You see, in endocytosis, it came in. And in exocytosis, it's coming out. Okay, so the cells also use exocytosis to secrete products, such as signaling molecules that need to be received by other cells. You got hormones, yeah, you got neurotransmitters. So now, because you know the direction stuff coming in. So now you can speculate. I can ask you on the exam, speculate what could be coming inside the cell. Give me example of stuff that can be endocytosed inside the cell. Give me example, please. What did we give an example of something that can come inside the cell? I already gave you two examples. So water is not endocytosis. Water comes through the tunnel. We're talking about, give me example of endocytosis, stuff that come inside the cell that is in a bubble. What's an example of that? Yeah, so the virus, the bacteria, they come inside what kind of cell? Yeah, phagocytosis, excellent. So inside the macrophage, inside the neutrophil, inside the immune white blood cell, we bring inside so we can fuse with the lysosome so we can kill it, destroy it. Very good. That's one example. What another example did I give you? So you are right in the sense that pinocytosis brings water, but water has channel known as aquaporin. But sometimes water mixed up with some other important solutes, we bring it inside in pinocytosis. So we do call it, yes, water does sometimes come in a vesicle, but not in a free form. It has its own gate, own tunnel to go through. But sometimes, because water is a versatile solvent, it dissolves many other things. So if it's in that form, then it doesn't go through the aquaporin. It goes through the pinocyte. So you're right, definitely. Water could be an example. What's another example I mentioned already? I didn't tell you cholesterol. That's how cholesterol come inside the cell in a clathrid coated fin. That's how you got LDL, low density lipoprotein that comes inside the cell. Now so we bring in the good stuff inside the cell. Sometimes we bring in bad stuff inside the cell to destroy it. Okay, now the issue. The issue, guys. The issue is this exocytosis. What is the purpose of? So if you look at exocytosis, the purpose of exocytosis is to expel material such as waste. So you have the cells also use exocytosis to secrete products such as signaling molecules that need to be received by other cells. You have hormones and neurotransmitters. So now we're going to secrete, some cells, they make hormone, like the nerve cell. They make a dopamine inside. Somebody got a Parkinson disease, they're always shaking, is because the dopamine is not secreted from the cell, from the substantia nigra. So some cells, they make neurotransmitter, so they need to send it out packaged in a balloon, in a vesicle, like that, like here. So this the blue inside here. What's an example of somebody blue inside? Neurotransmitter and also hormone. Insulin. Insulin come out the pancreas, exocytosis, balloon come out. Methods of transport, energy requirements, and types of material transport. So that you have to go through this chart, and we talked about diffusion. Diffusion is down concentration gradient, and it's affected by temperature. The more you heat up things, the faster diffusion is. The bigger the gradient is, the faster diffusion is, and the polarity of the molecule you want to diffuse in. Water is passive. Then active transport, then you got primary. What's the difference between primary active transport and secondary active transport? So Fatima, you mentioned for me active transport. We have two types. What is the difference between primary active transport and secondary active transport?
One need energy and the next one don't need energy, something like that.
Both need energy. It is the passive that don't need energy. But once you are active, you need energy. What's the difference?
What they're transporting?
No, we're talking about both of them are transporting different things. Yes. Primary, yes. So it uses chemical energy in the form of ATP. An example of that is sodium potassium ATPase. Requires ATP in order to move sodium against concentration gradient back out, 3 sodium back out, and 2 potassium back in. But secondary, it does use energy, but not in the form of ATP. It hitchhike. You're hitching the ride. Somebody come into school, you take a ride with them. You're not driving, but you take a ride with them. So in secondary active transport, you have sodium coming in the cell already, then you get glucose coming in with it. So glucose came in the cell at the expense of sodium already coming in. So when the water is falling in the waterfall, they put a rotor So the rotor blades spin because water coming in, and then inside they put magnet and electric wire, then you got electricity. So the electricity was based on the water hydraulic movement, this mechanical movement coming in, spinning the wire. Same thing here, the transporter, the rotor that brings in glucose spin because sodium coming in, not because ATP. In sodium potassium ATPase, it is ATP who makes the sodium potassium ATPase do the job. But in the glucose sodium transporter, it is sodium coming in, helping glucose to come with it. So the cells move large molecules into and out of the cells using bulk transport. That is bulk transport. So materials are moved into the cell using one of the forms of endocytosis based on the material being transported. Materials are transported out of the cell using exocytosis. That's 5.4. Let's now do 5.5. In 5.5, let's look at the cell surface interaction. In cell surface interactions, we're going to look at the appendages that allow for the movement of the cell walking, the cell crawling. So the extracellular matrix of the animal cells has a composition. What is the composition of the extracellular matrix? The extracellular matrix is a collection of material that is found in the extracellular space. Extracellular matrix is the collection of material that is found in the extracellular space surrounding the animal cells in multicellular organisms. So collagen is the most abundant protein and forms fibers. You need to know that what is the most abundant protein in the body? That is, what form does collagen come in? You know proteins come in primary, secondary, tertiary, quaternary. So what is the form that collagen comes in? I'm sorry? What is the form in which what is the form in which collagen comes in? Primary, secondary, tertiary, quaternary. Primary is the transcript, the nascent transcript that tells me, does my protein need to be folded quaternary? Does it need to be folded tertiary? Does it need to be folded secondary? Does it need to be folded primary? That's what it tells me. That's what the usefulness of the primary is. Primary structure has no usefulness except to tell me that we need to fold in what manner. So the primary transcript of collagen says that collagen is secondary. I told you last week that some jobs, all you need is secondary education, high school, that's enough. Some job, you gotta have college, associate. Some job, you gotta have bachelor. Some job, you gotta have master's. Some job, you gotta have doctorate. Some job, you gotta have postdoctorate. So the problem is, proteins have different structure based on their function. So collagen only needs secondary. Collagen and keratin. Where's collagen found? In the ligament. Where's keratin found? In the hair, in the nails. And then you have proteoglycans. The proteoglycans, these are carbohydrate containing proteins that are interwoven with collagen, where you have them all mixed up. So collagen is a secondary protein. The extracellular matrix of the animal cells has a function. So the matrix molecules connect to the extracellular surface of the cells. So the extracellular matrix holds the cells together to form a tissue, and it allows the cell within a tissue to communicate with each other, allows the cells in a tissue to communicate with one another. Okay. The extracellular matrix has important effects on the cell. So you have the binding of the matrix molecules to the cell receptors causes changes in the cell's activities through a sequence of events. So the matrix molecules bind to protein receptors on the extracellular surface of the cells. The receptor changes the shape of the actin filament just inside the plasma membrane. The conformational changes include chemical signals inside the cell that reach the nucleus. Then the transcription of specific DNA sections is turned on or off. Which proteins are made by the cell is going to change based on that, and then the cell activities are going to change. So we must understand that our blood viscosity must be under control. You don't want your blood to be too thin, and you don't need your blood to be too thick. Dangerous. If the blood is too thin, that means it's liquid. If it's too thick, that's very dangerous. So the blood clotting results from a series of events. So the cells lining a blood vessel are damaged. So damaged cells display a protein receptor called tissue factor. The tissue factor binds a factor within the extracellular matrix. This binding causes platelets to adhere to the damaged vessel wall. This stimulates the contraction of the adjacent smooth muscle cells in the blood vessel, and the blood vessel is going to constrict as a result, and the platelet is going to produce clotting factors. What are platelets known as? Band-aids. Are platelet cells? You know, we have white blood cells, the leukocytes, the eosinophils, the basophils, the macrophages, the monocytes. Then you have the red blood cells. You know, I had an exam, biology lab in another school, and I asked the students on the practical, I said, what kind of tissue is blood? What kind of tissue is that? Connective tissue. What kind of cells do we have in the blood? We have red blood cells. What else? White blood cells. What else? Plasma are not cells. I asked what are the cells in the blood. So the blood doesn't only have cells. Yes, it has plasma, it has water, it has electrolytes, it has proteins, it has lipids, it has carbohydrates. What are platelets? Are platelets cells? They're not cells. What are they? I'm sorry? What are they? They work as band-aids. That's their function. They function as band-aid. They stick to the site of injury to prevent blood leakage and blood loss. They stick there. But what are they? They're not, they are fragments of cells. Fragments, so they're not complete, they're just broken cells. Fragments of cells. Now, intercellular junctions. So now we need to pass from one place to another. We need to pass from one place to another. So if we need to pass, so you have intracellular junctions. So cells can also communicate with each other via direct contact or intercellular junction. So plants have one type of junction. These are known as the plasmodesmata. These are the plant cells. So for the name, from the name you tell, plasmodesmata. What are these junctions for? They are for communication. So sometimes, I went back home in my country, I visited my country, and I went to my friend's neighborhood. So my friend's neighborhood is very interesting. All the homes have like a door you can go from one house to another. Like let's say is a whole neighborhood, okay? So the neighborhood is not like here, like there's a fence and there's like a front yard, backyard. No, the houses are like all stuck together. There are streets, but the houses are stuck. But what I noticed is every house has a door bit. So let's say you have the houses are lined up one next to another. Each house has two doors that connect to each side of the other house. So you can, if you go inside one house, you can go through all the houses. That's just like the cell. Each cell has a junction that connects it to the next cell. So that's how they're able to pass electrolytes. That's how they're able to pass neurotransmitters. That's how they're able to pass all of these molecules from one place to another. You understand? So you can, as a molecule, you can cross through these junctions. Now you have to understand that not all the junctions, they're like doors. They allow you to go through. They're like tunnels. Some tunnels are like connections. They connect you. But some junctions, not every junction is a tunnel. All tunnels are junctions, but not all junctions are tunnels. Some junctions are not there to allow for passage of nutrients and molecules and cytoplasm and solutes. Some junctions are there to just staple the cells. The cells need to stick together. The cells are not sitting like you now. You have your own seat, you have your own boundaries. The cells are touching, stapled one another with these junctions. So some junctions are tight junctions, some junctions are gap junctions. They have that tunnel inside them. So where do we find the plasmodesmata? These are channels between the cell walls of the adjacent plant cells that connect their cytoplasms, allowing for direct communication via the transport of signaling molecules. The transport of materials like water, minerals, and nutrients from cell to cell throughout the plant. Okay, now if we look at the evolution of the connection, plasmodesmatas have evolved independently in different lineages of plants and algae. So they evolved repeatedly against the selective pressure of the cell wall to allow the plant cells to form effective multicellular organisms. These plasmodesmata have also affected plant virus evolution, and some viruses are small enough to pass through the plasmodesmata to infect and spread. So plants have evolved new ways to regulate and defend these openings, and viruses have evolved ways to continue to infect the plants. There you go. Intracellular junctions in the animals. These are tight junctions. Tight junction is a watertight seal between two adjacent animal cells that prevents the materials from leaking between the cells. These are the tight junctions, and they prevent the material from leaking between the cells. So you got to know those. You got to know the difference between a tight junction and a gap junction and a desmosome. A tight junction is a watertight seal between two adjacent animal cells that prevent material from leaking between the cells. So they are protective. They're protective. They prevent the movement of material. Then you have intercellular junctions in animals. These are tight junctions. You have proteins including claudins and occludins within the tight junctions, and they hold the cells together. These are protein complexes and proteins including the claudins and occludins within the tight junctions that hold the cells together. Intracellular junctions in the animals. So you have tight junctions, and these tight junctions are common in epithelial tissues that line the internal organs and cavities, and they comprise most of the skin. They line the urinary bladder and they prevent urine leakage into the extracellular space. Desmosomes act like spot wells between adjacent epithelial cells. These junctions maintain the cells in a sheet-like formation in stretchable organs and tissues, like the skin, the heart, and the muscles. You also have intercellular junctions in animals. These are the desmosomes. So the plasma membrane proteins are called cadherins that connect to keratin, which is a type of a cytoskeletal filament, to create the desmosomes. So desmosomes are created by these cadherins. You see, these are a type of intracellular junction in the animals, and they act like spot welds between the adjacent epithelial cells. These junctions maintain cells in a sheet-like formation. So they hold the cells in place, providing structural tensile integrity. The plasma membrane proteins are called cadherins, and they connect to keratin, which is a type of cytoskeletal filament, to create the desmosomes. Gap junctions are protein-lined channels between the adjacent cells that allow for the transport of the ions, nutrients, and other substances that allow the cells to communicate. They are functionally like plasmodesmata in the plant cells. Intercellular junctions in the animals. These are the gap junctions. You see the tunnel? So that's cell number one. You see where my mouse is? That is cell number one. It allows the passage through this tunnel. So that's a hydrophilic channel. Allows the passage through this tunnel. So a gap junction forms when 6 connexin proteins come up together on the plasma membrane to form a connexon. A connexon in one cell aligns with a connexon in an adjacent cell. So that shows us here, now the animal cells communicate via their extracellular matrices. So when protein receptors on an animal cell membrane surface bind to an extracellular matrix substance, this results in changes in the cell's internal activities. The animal cells may be connected to each other via tight junctions, known as desmosomes, and gap junctions. So a tight junction is a watertight seal between two adjacent cells. A desmosome acts like a spot weld, and a gap junction is a channel between the adjacent animal cells. Plant cells are connected and they communicate with each other via the plasmodesmata. And while gap junctions and plasmodesmata are functionally similar, their structures are quite different. Okay, so that was chapter 5. So what are we gonna do after chapter 5? 7. Excellent. I was waiting somebody gonna say 6. So now we're going to go into the bioenergetics. And how do we fuel these biochemical processes? One of the ways in which we can get energy was the ATP. That's a type of energy. Energy in the form of adenine triphosphate, nucleic acid energy. Now, in addition to the ATP, we also learned about the secondary active transport. In that one, we've seen that we shuttle one molecule across the plasma membrane with the help of an original initial electrolyte that is going to move down concentration gradient. Then we use it to actively move another molecule. So plants and animals convert energy from food into energy cells that can use as ATP. So you have to understand that cellular respiration is all about being able to metabolize. It's about being able to perform metabolism and to be able to generate energy. So cellular respiration extracts energy from glucose bonds and converts it to ATP. So do we only get energy from glucose? So glucose is one type of macromolecule. There is sugars. Glucose is a sugar. So do we only rely on sugars for energy? Of course not. We rely on sugars. We rely on proteins. We rely on fats. Excellent. And in addition to fats, we rely on nucleotides like ATP. Excellent. So the energy in the living system involves oxidation, reduction, reaction. These are known as redox reactions. So the oxidation strips the electrons and reduction adds electrons. Oxidation strips the electrons while reduction adds the electrons. It transfers energy via these electron shifts. We already talked about Leo says. What did we say? Leo says. We mentioned in the past. Leo says lose electron oxidation. Leo says GER, gain electron reduction. We didn't say that. So Leo lose electron excitation. GER, gain electron reduction. So oxidation strips the electrons and reduction adds electrons. Oxidation strips electrons, reduction adds electrons. So the energy in the living system transfers energy via these electron shifts. So in oxidation, you lose an electron, and we see that we oxidize methane to get methanol. Then we oxidize formaldehyde. We oxidize methanol to get formaldehyde. Then we oxidize formaldehyde to get formic acid. Then we oxidize formic acid to get carbon dioxide. Electrons and energy. The removal of electrons, oxidation, results in a decrease in potential energy in the oxidized compound. So the more you're oxidized, the less energy you have. So when you're oxidized, you lose electrons. Remember, I talked to you in the past that cells, they do not exchange proton plus. Why the cells cannot give each other proton? Because proton is already locked inside the nucleus, right? Cannot come out, cannot break the nucleus. But who's already came out spinning around the nucleus? The electron. So they can give each other electron. Us, we give each other money. So if your bank account has a plus 100, if your bank account has plus 200, you're happy. But if your bank account has minus 200, you're sad. But the cell, if the cell has minus 200, it's really happy because it doesn't participate in dollar exchange, it participates in electron. So once you give it a minus, it's happy, it's rich. If it has a minus, it means it has electron. But if it has a plus, that means it lost an electron. So if you oxidize something, you strip it away. You strip electrons away from it. So that's bad. But when you gain electrons, you make it happy. So now you result in an increase in the potential energy in a reduced compound. So high energy electrons are going to fuel the cells. So the cells use high energy electrons to incrementally transfer energy. So the electron shuttles bind and carry high energy electrons. An example of that is the NAD plus and FAD plus. So you can tell NAD plus, is that oxidized or reduced? Plus means what? Plus means gained or lost energy. It means lost. So we said Leo, lose electron, oxidation. So how you tell me NAD plus, lose electron is reduced? Is NAD plus oxidized or reduced? Oxidized. Now, once it gains an electron, does that make it oxidized or reduced? Reduced. Now, the other thing you need to learn is that even though you know who's oxidized, you know who's reduced, you're never going to be asked who's oxidized, who's reduced. They're going to ask it to you in a different way. They are going to ask you who's the oxidizing agent, who's the reducing agent. Now, you got to be careful because the oxidizing agent is the one who's reduced. So do not say the oxidizing agent is oxidized or the reducing agent is reduced. They cannot be two in one. This is very, very, very important. You cannot be oxidized and the oxidizing agent at the same time. You cannot be reduced and the reducing agent at the same time. If you're oxidized, that means you're the reducing agent. If you are reduced, that means you're the oxidizing agent. Are we clear? Are we clear? Anybody has a question? So now when you look at NAD+, is NAD+ the oxidizing agent or the reducing agent? To answer a question like that, start with Leo says Ger. When you find out from Leo says Ger, who's oxidized, who's reduced, the second part of the formula is, if you're oxidized, that means you're the reducing agent, because you cannot be two in one, remember? And if you're reduced, you cannot be the oxidizing agent. So the first step, Leo says Ger. Find out who's the oxidized, who's the reduced. Then after you get it, it's easy now. Because if you're oxidized, what kind of agent are you? Reducing agent. If you're reduced, what kind of agent are you? Now look at NAD+. Is it the oxidizing agent or the reducing agent? It's the reducing agent because it is oxidized. It lost an electron. Very good. So ATP is the energy currency of the cell. We store and transfer energy in the form of ATP. by removing the last phosphate group from ATP. Net releases energy. So phosphate binding to another molecule activates the energy. Phosphate binding to another molecule activates energy. So an example is sodium-potassium pump. So sodium-potassium pump is very interesting because ATP alters the structure of the integral protein that works as a pump, changing its affinity for sodium and potassium. This is how the cell performs work, pumping ions against their electrochemical gradient. Adenosine monophosphate is part of ATP. Adding a phosphate group to adenine monophosphate produces ADP. Adding a phosphate group to ADP produces ATP. So adding a phosphate group requires energy. So dephosphorylation is from the name, dephosphorylation. Dephosphorylation means what? Means you remove it by releasing it. You release a phosphate group from ATP to release the energy. Phosphate groups are removed through hydrolysis, adding water, H2O. So you see adenine triphosphate. I want to tell you something. If you have one nitrogenous base, can you identify the nitrogenous base here? That is the nitrogenous base. What is that in the middle? That's the nitrogenous base. What is that? This is the nitrogenous base. And what is this? That's the ribose sugar. And one phosphate here, if you have 3 adenosine, ribose, and a phosphate, that's called your nucleoside. But if you add these 2 phosphates, it's going to be triphosphate, because you remember you had 1 phosphate, that's adenine monophosphate. If you have 2, that's adenine diphosphate. If you have 3, that's adenine triphosphate. So there we go. Remember we talked about hydrolysis. Hydrolysis comes in to break the bond. So how did we go from ATP to ADP? We went through hydrolysis. We hydrolyzed one of the phosphate bonds and we ended up with adenine diphosphate. So hydrolysis breaks down the complex macromolecules, and the hydrolysis of ATP produces ADP and phosphate, and ATP is regenerated by adding phosphate to ADP, and ATP links catabolism to anabolic pathways. There you go. This is, if you have adenine triphosphate, removing 1 phosphate, that's going to release energy. And now you're going to have adenine diphosphate. Then adding another phosphate is going to restore us back from ADP into ATP. From ADP into ATP. Now, if we look at phosphorylation, enzymes form intermediate complexes to allow substrates and ATP to interact during reactions. So enzymes form intermediate complexes to allow substrates and ATP to interact during the reactions. So phosphorylation happens when adding a phosphate group by using intermediate complex to transfer ATPs third phosphate group to substrate. So the driving force for turning cellular processes on and off is phosphorylation. Now we have substrate level phosphorylation. So there are chemical reactions of adenine diphosphate that produce ATP through catabolic reactions. So substrate level phosphorylation is when the phosphate group is removed from the intermediate reactant and is attached to ADP, producing ATP. So we need a donor. We need a donor that's going to donate for us phosphate. We're going to take phosphate from the donor and we're going to add it to adenine diphosphate. What is that going to give us? Excellent, because we had ADP, adenine diphosphate. This adenine diphosphate, if you add a phosphate group to it, it's going to become adenine triphosphate. Where are we going to get the phosphate group from? We need a donor. Who's the donor who's going to give us the phosphate group? Excellent. That's the intermediate or the substrate. Excellent. Very good. Oxidative phosphorylation. Most of ATP is generated from chemiosmosis. In chemiosmosis is a process of ATP production in cellular metabolism. So oxidative phosphorylation is very important because that's an example where aerobic respiration is going to come in. Why do we call it aerobic respiration? Because we utilize oxygen. So most of the ATP is generated from chemiosmosis. And chemiosmosis is a process of ATP production in cellular metabolism where you have oxidative phosphorylation and production of ATP using chemiosmosis. You see, chemiosmosis is the process of ATP production in cellular metabolism, and there is oxidative phosphorylation, that is the production of ATP using chemiosmosis. So we have learned that ATP is the energy currency of the cell, stores energy, transports it for endergonic reactions. It loses the phosphates to become ADP, AMP when used, and ADP is regenerated to ATP using glucose catabolism energy. Phosphates are attached to substrates during phosphorylation, and there are two ATP regeneration processes. The first one is substrate level phosphorylation. That is when we add directly to ADP phosphate from a donor, an intermediate substrate, as Devin. Your name is Devin? Yeah, Devin nicely said it's a substrate. It's an intermediate. You see here, this is the substrate. This is the substrate. The intermediate substrate is going to donate the phosphate. Then we have the oxidative phosphorylation, this chemiosmosis, where you have the ATP synthase enzymes and the electron transport chain are embedded in the inner membrane. Now we have to examine glycolysis. Glycolysis is, remember we said metabolism can be performed in different nutrients. So we said how many types of nutrients do we have? We have carbohydrates, we have proteins, we have fats, lipids, and we have nucleic acids. If we're going to get ATP from sugars, what is that known as? Glycolysis. Glycolysis is the pathway that enables us to get ATP from sugar. So it's the first step in breaking down glucose to extract energy for cellular catabolism. So what are we going to do to glucose? Are we going to reduce or oxidize glucose? If we're going to get energy out of glucose, are we going to oxidize it or reduce it? We're going to oxidize it. Very good. So glycolysis is an anaerobic, meaning if you hear anaerobic, what does that mean? It means we use energy, we use oxygen, or we do not use oxygen. We do not use oxygen. So an anaerobic with oxygen process that occurs in the cytoplasm of all the cells. So glucose enters the cells via secondary active transport. Transport that takes place against the glucose concentration gradient. So the cell already has a lot of glucose inside. So to bring the cell in, we need a force that allows the entry of the glucose. The glucose transporter, the GLUT, we talked about those. Glucose transporters, the GLUTs, these are a group of integral proteins that assist in facilitated diffusion. They assist in the facilitated diffusion. There we go. So we see that in glycolysis, we see that we add ATP, and when we add ATP to glucose, we invest 2 ATP molecules, and then we get 4. So it's like, let's say you invest $2,000, but you get $4,000. How much did you make? So that's exactly what happens in glycolysis. Please examine this figure carefully. That's our glucose. So we take glucose. How many carbons are in glucose? 1. We said every intersection that has no letter on it, that means it's an intersection. That's not an intersection. That's a hydrogen. If you see something like that, that's a hydrogen. But an intersection is a carbon. That's 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. These are 6 carbons. Now, if I break down glucose, what do I break it into? It has 6 carbons. What do I break it down into? 2 carbons that each have how much? 1, 2, 3. Because it's a 6 carbon molecule, I divide it into 2 3 carbon molecules. Do you agree with me? If it's a 6 carbon molecule, I divide it into 2 3 carbon molecules. Is that clear? Each of the 3 carbon molecules is called what? If I break this 6 carbon glucose into 2 3 carbons, each 3 carbon molecule is called what? Pyruvate. Yes, it's called pyruvate. So glycolysis is when I break down a 6 carbon glucose into 2 3 carbon pyruvates. What do I invest? 2 ATPs. What do I get total? 4. What do I get as a net? What do I get as net? 2. As net, you get 2. Total, you get 4. Investment, you get how much? 2. Excellent. So you get 2 ATPs and you get 2 NADHs. What are these NADHs? These NADHs are oxidizing agents. They are oxidizing agents because they are reduced. They have potential. They have electrons to lose. They have electrons to later on oxidize. That's why we call them the oxidizing agents. So glycolysis gives me 4 total ATP, 2 net ATP, and how many NADHs? 2 NADHs, and how many pyruvates? Look, it's not a trick. Look. 2. You got to know all of that. That glycolysis is when I break down and I oxidize a 6-carbon glucose into 2 3-carbon pyruvates, and I get a total 4 ATP. After I invested 2 ATPs, that's going to come out to a net 2 ATP. All that you got to know. Very important for the glycolysis. There are 2 distinct phases. The first phase is the investment phase. The second phase is the production. So first you invest, then you get back. So it traps glucose in the cell and uses energy to modify it so that the 6-carbon sugar molecule can split into 2 3-carbon molecules. It extracts energy from the molecules and stores it in ATP and NADH. Each step is catalyzed by different enzymes. So the first half of glycolysis is the energy requiring step. The first step is catalyzed by hexokinase enzyme. It phosphorylates glucose to give us glucose 6-phosphate. That's the first ATP we invested. That's where we get the donor, our first donor. Remember we said we need a phosphate donor? So we got to use ATP as a donor to give us phosphate so we can add it to the glucose. That's called glucose 6-phosphate. That prevents the phosphorylated glucose molecule from continuing to interact with the glucose transporter proteins, trapping it inside the cell. So you know that glucose is going to leak out. It's going to leak out down concentration gradient. Is there more glucose inside the cell or outside the cell? Is there more glucose inside the cell or outside the cell? Most of the time. It's mostly inside the cell because we store glucose as glycogen. It's only when you eat the pancake, when you eat the sandwich, now you have more glucose in the blood. But usually there's more glucose inside the cell. So now, if you want to bring in glucose inside the cell and perform glycolysis, if you brought it in, it's going to leak out because there's more glucose in, down concentration gradient. So that's why we tag it with phosphate. That's the first step. We have ATP, so ATP can donate phosphate. We add phosphate to glucose. Glucose is not going out anymore. That's the first investment phase. Why do we use ATP in the first step of glycolysis? It is to add phosphate from the ATP to glucose to prevent glucose from leaking out. Step 2 is catalyzed by an isomerase enzyme. It's an enzyme that will make a rearrangement. The isomerase enzyme is going to catalyze the conversion of a molecule into one of its isomers. It converts glucose 6-phosphate into fructose 6-phosphate. It allows splitting into 2 3-carbon molecule. Step 3 is catalyzed by phosphofructokinase. That is a rate limiting enzyme. So phosphofructokinase, whenever you hear kinase, what should you think of? Whenever you hear kinase, what should you think of? If you hear the word kinase, what does that tell you? What does ACE tell you? It's an enzyme. So take all the given information. That's an enzyme. Kinase is an enzyme. The other problem, what is kyne? Kyne means it's gonna add a phosphate group. It's gonna phosphorylate. Phosphorylate. Kinase is going to phosphorylate. Kinase is going to phosphorylate. So phosphofructokinase is going to phosphorylate fructose 6-phosphate to fructose 1,6 bisphosphate using ATP. So you know isomerase rearranged for us glucose into fructose. And remember, when we added that first phosphate to prevent glucose from leaking out, that's one phosphate. Because remember, you have glucose that has 6 steps. One of the steps is phosphate, was added to the first. Remember, it has 6 carbon. One carbon has a phosphate. Then we use phosphofructokinase to add phosphate to the other side. So now we're even. That's why they call it fructose 1,6 bisphosphate, meaning that it has phosphate in both sides. So that when I break it in half, I got my 2 pyruvates, each one 3, each one has a phosphate. A high energy phosphates destabilize fructose 1,6 bisphosphate. It is catalyzed by aldolase enzyme that cleaves fructose 1,6 bisphosphate into 2 3-carbon molecules, DAP and G3P. It is catalyzed by isomerase that transforms DHAP to G3P. Is an investment of 2 ATP molecules to break down 1 glucose molecule. Second half of glycolysis is, and these are the energy releasing steps. So far, glycolysis costs 2 ATP and produces 2 G3Ps. Now the energy will be extracted from the glucose triphosphate molecules to produce 4 ATPs. That oxidizes glucose 3-phosphate by transferring the electrons to NAD plus to form NADH. It phosphorylates the oxidized sugar to 1,3-bisphosphoglycerate, and it depends on the availability of NAD plus. So NADH must be continually oxidized back to NAD plus. This is catalyzed by the phosphoglycerate kinase. So that's the enzyme that's going to catalyze the step. And the remaining phosphate group moves from the 3rd carbon to the 2nd carbon, producing 2-phosphoglycerate. Step 10 is catalyzed by pyruvate kinase. That is the rate limiting enzyme. There is a substrate level phosphorylation, produces a second ATP. And you have to understand that this enzyme PEP is going to be converted into pyruvic acid. And then pyruvate, that is the salt that is made from pyruvic acid. It has 3 carbons. You see how we start from glucose? And these are all the steps of glycolysis. So when glucose comes inside, so you know you drink milk, that's lactose, made up of galactose and glucose. You eat sucrose, that's the sugar cane or the table sugar, that is made up of glucose and fructose. You eat maltose, that's the oatmeal, that is made up of glucose and glucose. So once glucose is inside the body, it's gonna have to go down this 5 steps. The first step, converting glucose into glucose 6-phosphate. Second step is converting glucose 6-phosphate into fructose 6-phosphate. Then converting fructose 6-phosphate into fructose 1,6-bisphosphate. Then fructose bisphosphate aldolase is going to be converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So these are the 10 steps that culminate in giving us pyruvate. So these are the outcomes of glycolysis. They produce 2 pyruvate, 4 ATP, and 2 NADHs from 1 glucose. The net gain of 2 ATP and 2 NADH after the investment phase, that is the first half of glycolysis. An example would be the critical ATP source for anaerobic red blood cells that cannot perform aerobic respiration. But if we look at aerobic respiration, that is when we utilize oxygen, converting energy in the presence of oxygen. And if there is no glycolysis, the cells cannot maintain the sodium-potassium pumps, and they will die. So the last step will not occur if pyruvate kinase is insufficient. So glycolysis is the first pathway that is used to break down glucose and extract energy. It is an ancient metabolic pathway found in nearly all the organisms. They consist of 2 parts. The first part breaks down the 6-carbon glucose into 2 3-carbon sugars, investing 2 ATPs. The second part extracts the energy and products and produces 4 ATP and 4 NADHs. And lastly, the ATP is generated by substrate level phosphorylation, not by oxidative phosphorylation. So we're done with the substrate level phosphorylation. Now we have to look at the oxidation of the pyruvate and the citric acid cycle. So we went in this stepwise journey from glucose into pyruvate. And we got net how many ATPs? We got 2 net ATP. Do we stop there in pyruvate? No, we take pyruvate and go more. Do you make dough? You bring the flour and you bring your milk and you bring the eggs and you bring the butter. You mix them all up. Now you have dough. Do you throw the dough in the garbage, or you go further, you add some, you do some more with it? So the same thing. We made pyruvate with 2 ATPs, but that's not the end. We can continue now. So now we're going to continue. What are we going to continue to? We're going to go to the citric acid cycle. So the citric acid cycle comes immediately right after glycolysis. Very good, glycolysis. The oxidation of pyruvate and the citric acid cycle. It occurs in the mitochondria of eukaryotic cells under aerobic conditions. It occurs in the mitochondria of eukaryotic cells under aerobic conditions. So where does the citric acid cycle happen? It happens in the mitochondria, and it must be happening under the presence of oxygen. If there is no oxygen and there is pyruvate, there will never be oxidative phosphorylation. You have to know that. Even if there is a mitochondria, there is pyruvate, but if there is no oxygen, there will never be oxidation of pyruvate in the citric acid cycle. The citric acid cycle will shut down. Citric acid cycle requires oxygen. Pyruvate from glycolysis is converted into an acetyl group. There's a small molecule made of 2 carbons, 3 hydrogens, and 1 oxygen. 3 carbons, 3 hydrogens, 2 carbons, and 1 oxygen. So the acetyl group then is picked up and activated by coenzyme A. The result is coenzyme A is a combination of an acetyl group from pyruvic acid and coenzyme A. So you see the acetyl group is then picked up and activated by coenzyme A, and the result is acetyl coenzyme A. So you must understand that the pyruvate from the glycolysis is converted into acetyl group, a small molecule of 2 carbons, and then that acetyl group is picked up and activated by coenzyme A to get acetyl-CoA. So you have to understand that this 3 carbon pyruvate that I get at the end of, what's the name of the process that gives me pyruvate from glycolysis, from glucose? Glycolysis. So now I have my pyruvate. I take pyruvate, but pyruvate has 3 carbons. I break down pyruvate from 3 carbons into how many carbons? 2 carbons. What is that called now? It used to be called pyruvate when it was 3. When it was dropped down to 2, what is it called now? I can see it right there. Acetyl. So now this acetyl is going to be taken and we're going to add to it coenzyme A, and we're going to end up with acetyl coenzyme A, acetyl-CoA. Now let's look at what acetyl-CoA is going to do. Acetyl-CoA is a combination of the acetyl group that we took from the pyruvate, breaking down 3 carbon pyruvate into 2 carbon acetyl, and coenzyme A. So the pyruvate is the product of glycolysis. Do you see pyruvate? That's the product of glycolysis. It must go through a 3-step process to convert to acetyl-CoA. So the conversion of pyruvate to acetyl-CoA occurs while pyruvate is translocated across the mitochondrial membrane. When it's translocated across that's the breakdown of, so now let's look at the breakdown of pyruvate. So the carboxyl group is removed from pyruvate, releasing carbon dioxide, and that creates 2 carbon hydroxyethyl group that is bound to pyruvate dehydrogenase. That is a multi-subunit enzyme embedded in the mitochondrial membrane that catalyzes the conversion of pyruvate into acetyl-CoA. This step happens twice to remove 2 of the 6 carbons. Hydroxyethyl group is oxidized to an acetyl group. So the electrons are transferred from the NAD plus to form NADH. Then the enzyme bound acetyl group is going to be transferred to coenzyme A, forming acetyl-CoA. So that's our pyruvate. An oxidation reaction is going to cause decarboxylation. We removed carbon dioxide. You see the decarboxylation? Why do we call it decarboxylation? Because we take out carbon dioxide. We used to have 3. Remember, in pyruvate we had 1 carbon, 2 carbon, 3 carbon. But now we're going to remove carbon with 2 oxygens. That's going to be carbon dioxide. So now we have 2 carbons. And that's going to be acetyl. And we're going to add to it this sulfur group that has the CoA. That's known as acetyl-CoA now. Now we're going to go from acetyl-CoA to carbon dioxide. Acetyl-CoA delivers its acetyl group to the 4-carbon oxaloacetate to form citrate. This acetyl-CoA is going to bind this acetyl-CoA This acetyl-CoA delivers its acetyl group to the 4-carbon oxaloacetate to form citrate, the 6-carbon molecule with 6 carbon groups. This initiates the citric acid cycle to extract the remaining energy and releases carbon dioxide. The citric acid cycle is a series of enzyme catalyzed chemical reactions of importance in all living cells for extraction of the energy from the carbohydrates. You have the tricarboxylic acid cycle and the Krebs cycle. The citric acid cycle takes place in the mitochondria, which is a closed loop with the last part regenerating compounds used in the first step. There are 8 steps total. There is reduction, oxidation reactions. There are dehydration, hydration, and decarboxylation. In the end, what is it going to give us? It's going to give us 1 ATP, and it's going to give us 2 carbon dioxides and 1 NADH and FADH2. These are oxidizing agents. The aerobic pathway that produces little ATP and does not consume oxygen directly. You see, this is the citric acid cycle. Do you see the citric acid cycle? The citric acid cycle begins with what? Begins with pyruvate being converted into acetyl-CoA. This is a very important enzyme. You don't have to memorize all the enzymes here. But the very important one is the one that takes us from pyruvate to acetyl-CoA. Because you know, if it wasn't for the acetyl-CoA, you can never enter the citric acid cycle. You cannot enter the citric acid cycle as pyruvate. You need to be converted from 3-carbon pyruvate into 2-carbon what? What is it called? Acetyl-CoA. Acetyl-CoA. So this acetyl-CoA can now enter, and since it has 3 carbon, it will find citrate. You know citrate from orange juice? Citrate has how many carbons? 4. 4 from citrate, 2 from acetyl-CoA. How much does that come out to? And then the cycle is going to spin, and then it's going to give us, or these are our products. See the products? 2 carbon dioxide, 1 GTP ATP, and there are reduced electron carriers known as NADH and FADH2. Okay. These are the steps of the citric acid cycle. So you don't have to memorize these steps. Just understand that the significance is that pyruvic acid is converted to acetyl-CoA. There is a condensation reaction that combines 2-carbon acetyl group with a 4-carbon oxaloacetate to form 6-carbon citrate. It is an irreversible, highly exergonic rate that is controlled by ATP levels. Citrate loses 1H2O, becoming cis-aconitase, and then the cis-aconitase is going to gain another to become an isomer, known as D-isocitrate. Then diisocitrate is going to be oxidized to give us alpha ketoglutarate. So it's about 4 or 5, all these 7 steps, but they're going to, in the end, give us these products. We're going to have 2 carbon dioxide molecules that are released per turn of the cycle. There are 3 NADH and 1 FADH2, and that makes the cycle amphibolic, meaning that it is both catabolic and anabolic at the same time. So in the presence of oxygen, pyruvate from the glycolysis is converted to acetyl-CoA. And the cycle is a series of a combination of redox and decarboxylation reactions, amphibolic. The electron transport chain is a step that comes after the citric acid cycle. So the ATP from glycolysis and citric acid cycle derived by moving the electrons. So the electrons move through a series of consecutive cascade of electron carriers in a reduction oxidation reactions. So the hydrogen protons accumulate in the mitochondria and they pass through the ATP synthase. ATP synthase is an enzyme that uses the energy released by the hydrogen proton moving down the concentration gradient to produce ATP. So the synthase is the motor, the rotor that is going to generate for us ATP. It's a protein enzyme that uses the energy released by the hydrogen proton moving down the concentration gradient to produce ATP. For ATP synthase, phosphorylates ADP to give us ultimately ATP. So you see these are successive proteins that generate for us a proton motive force. This proton motive force is going to allow the influx of hydrogen protons down the ATP synthase, and it's going to create this rotor and the hydrogen proton. This hydrogen proton is going to move in, and this, so you must understand this step here. You see where my mouse pointing? This motor, this rotor is going to spin. Can everybody see my mouse there on the monitor? You see this motor, this rotor here? This rotor is going to spin because hydrogen proton is flowing down, influx, where the arrow is pointing from the hydrogen proton is floating down. When it floats down, it's going to spin, and then ATP is going to synthesize. This ATP synthase, that means it's an enzyme. So this ATP synthase is going to spin, and it's going to add phosphate to adenine diphosphate to give me adenine. Triphosphate. So the electron transport chain is the last component of the aerobic respiration. It is the only part of glucose metabolism that requires atmospheric oxygen. This one requires oxygen. But remember, we have all of these complexes doing what? You see where my mouse pointing? You have all these complexes pumping out hydrogen proton, pumping out hydrogen proton, pumping out hydrogen proton, pumping out hydrogen proton. So there is so much hydrogen proton right here building up. So because you have so much hydrogen proton out, it's going to flow down through the ATP synthase. So our job is to push so much hydrogen proton. That's known as electropositive or a proton motive force. Then the hydrogen proton is going to influx through the ATP rotor, the ATP synthase, and the ATP synthase is going to rotate This ATP synthase motor is going to rotate, and while it's rotating, it's going to add a phosphate to this adenine diphosphate ADP, converting it into adenine triphosphate, and that's what we want. You get it? So these are the different complexes that participate in, we're not going to go through all of them, but this is just shows you the chemoosmosis is where the free energy from the redox reaction is used to pump hydrogen ions across the mitochondrial membrane. So chemoosmosis, you see right here, there we go, that's chemoosmosis. So I have oxidation reduction, oxidation reduction, oxidation reduction. So these oxidation reduction movements are going to pump out or pump in hydrogen proton. What do you see here? No, it's not pumping in. It's pumping in the last step. All of these guys, oxidation reduction, because remember, lose electron oxidation. So this one lost electron, this one gain electron. This one is going to lose electron, this one is going to gain electron. This one is going to lose electron, this one is going to gain electron. Does this one have anything to do with electrons? So it's these here, these four, complex 1, complex 2, complex 3, complex 4, these are the ones who do the redox. So why are they doing redox? So they can pump out the hydrogen proton, pump out hydrogen proton, pump out hydrogen proton. When hydrogen proton is pumped out, who's going to bring it in and spin to give us ATP? You have to look in the monitor where my mouse is. So when I ask you a question, you're not all confused. I'm talking with my mouse here. You see this mouse? I'm doing chemoosmosis. You see where the electrons are here. So I'm going, complex 1 is going to donate electron, it's going to be oxidized. This one is reduced. You get it? Yeah. This complex 2 is going to get oxidized. This one is going to get reduced. This one is going to get oxidized. This one is going to get reduced. And in the last step, we make water. But while we were oxidize, reduce, oxidize, reduce, oxidize, reduce, what was happening in the meantime for hydrogen proton? It was getting all pumped out. So when it's all pumped out, there is so much hydrogen proton here, it's going to diffuse down like the waterfall. When the waterfall falls down, it's going to spin the turbine, the turbinate. So when it's going to spin the synthase, that's going to make for me ATP. So that's chemoosmosis here. So chemoosmosis is where the free energy from the redox reaction is used to pump the hydrogen ions across the mitochondrial membrane. So it creates an electrochemical gradient with a higher concentration of protons on one side of the membrane, and ATP synthase is going to allow the protons to diffuse back through using the energy to phosphorylate ADP to form ATP. It is used to generate 90% of ATP that is made during the aerobic glucose catabolism. It is also used in light reactions of photosynthesis, known as we will see that in the photophosphorylation next chapter. So it looks like next week, chapter 8 is not going to be on the exam. So it looks like next week you don't have chapter 8 homework due. I'm going to extend that to the following week. So we're going to push off the chapter 8 to next lab exam. So our lab exam next week is going to stop at chapter 7. Today we will finish at chapter 7. So all your homeworks are going to be due for chapter 7. You're going to have your Canvas homework for chapter 7. You're going to have a study guide, but you don't have to submit it. Just study it. You don't have to turn it in anymore. Just use it for studying. But you're going to have the virtual labs. Those virtual labs on McGraw-Hill Connect, you're going to do those. So next week, what do you have? You have McGraw-Hill homeworks up to chapter 7. You have a Canvas homework. You have a lab exam Canvas homework, like you had a midterm exam homework. You're going to have a lab exam homework, and you're going to have the McGraw-Hill Connect. and I'm gonna extend chapter 8 McGraw-Hill to next week. And from last week, if you did not do McGraw-Hill, I extended it for you to next week. So all they're gonna be due next week. And I'm gonna push the 8 to the next week. I'm gonna type all of that for you on Canvas so you don't come next week. I didn't know. I'm gonna show you it's right there. You're gonna get a 0. You're gonna get a 0. No problem. Okay, sure. I appreciate you too. Let's finish. We're not done. We're not done. We're not. That's not. I said when we finish 8. We're not. We are 7. We are 7 point. Oh, please. We are 7 point. That's 7.3. You have 7 point. That's 7.4. No. There's 7.5, 6, and 7. Hold on. I'll give you a break. I'll give you a break when we finish this. Then we come back. We do 7.5, 7.6, 7.7. Then you go home. Okay. No. I gotta do that. Okay. So this is what I was saying. So if you work with me, we're gonna go faster. But if you're sleeping and I'm asking questions, that's gonna hold us back. So we participate. We're gonna move further. So what we say? We said that we need to generate a proton motive force. We need hydrogen proton to be pumped in order for us to spin this rotor. In order for us to spin the rotor. So we're gonna have the ATP synthase, and that's gonna. So we're gonna have an ATP yield. So the number of ATP molecules generated is gonna vary based on factors like the species, the electron shuttles, the uncoupling proteins, and intermediate compounds are also used for further purposes. We said that sugars, other than glucose, are fed into the glycolytic pathway. Pentose sugars, non-essential amino acids, and lipids are made from intermediates in the glycolysis and or the citric acid cycle. In living systems, glycolysis and the citric acid cycle extract about 34% of the energy from glucose, with their remainder released as heat. Okay. So electron transport chain is the portion of the aerobic respiration that uses oxygen as the final electron acceptor. So the electron transport chain is composed of 4 large protein complexes embedded in the inner mitochondrial membrane and 2 small electron carrier molecules that shuttle the electrons between them. So the NADH and FADH2, these are oxidizing agents. Oxidizing agents meaning that they perform, they will undergo, they are reduced, but they are willing to undergo oxidation in order to reduce somebody else. They donate high energy electrons that pass through a series of redox reactions with some energy used to pump the protons across the membrane to establish a gradient that contributes to chemiosmosis. So the electrons gradually lose the energy as they are passed down the chain until they reduce the oxygen to form water as the final electron acceptor. The end products of the electron transport chain are water and ATP. Okay, let's take a break and let's come back in 45 minutes, and then we will spend